Mainly it aims to develop an inbred lines which are used in the hybrid breeding. And today we will see, like, up to this class we have studied like what is heterosis, what is the genetic basis of heterosis, and we also studied like how these heterosis can be exploited in many ways. Either the partial heterosis can be exploited in synthetics and composites or population improvements, and either you can complete exploitation of heterosis by hybrids. And today we are going to discuss how exactly hybrid breeding will takes place. Like we have seen types of hybrids and different but I told about types of hybrids and how to maintain the hybrids and how to cross them and how to do what are the precautions we have to take and what are the conditions we have to maintain we discussed. And I have not discussed in detail like how to develop an inbreds. One methodology I have discussed, double haploid, and rest other methodology we are going to discuss. Before that, I will summarize the law points which I have discussed. The nature of cross-pollinated crops, we know that they are heterozygous, heterogeneous, and they contain high mutation load than inbreeders. Why it is so? Why they contain high mutation load than the inbreeders? What is the reason? Why inbreeders having low mutation load?
Sir, in cross-pollinated crop, the recessive mutants exist in the population, sir. Whereas in self-pollinated crop, they already get exposed to the natural selection and they will get lost from the population, sir, generally.
That is the reason why the high mutation load than the inbreeders. In self-pollinated crops, the mutations which are unfit for the survival of the particular crop, it is get it's not selected through in the evolution. They lost during the evolutions. That's why the mutation which are determinantal or which are it is not good for the performance of the particular line of crops. It get eliminated during the course of evolution. That's why. But in cross-pollinated, they maintain in a heterozygotes. Okay, the recessive mutations which are uh, having lethal effect, they are maintained in the heterozygotes, so that they are having high mutation load than compared to inbreeders. And it is major another important nature of cross-pollinated crops is inbreeding depression. Because of this, the inbreeding depression, whenever you enforce a continuous selfing in the cross-pollinated crop, there will be high inbreeding depression. Especially, you should remember the classification: high inbreeding depression crop, moderate inbreeding depression crop, low inbreeding depression crops. Okay, then breeding procedures is mainly achieve the maximum efficiency by the combining, okay, like combining selfing procedure and also outcrossing that we have seen in population improvement approaches. First we will self, then you outcross, then you will maintain heterozygosity. At the end we are developing an heterozygous individual, the population of heterozygous individual we are going to release. In the case of hybrid breeding also, we are releasing an hybrid as a variety, means it's a F1 heterozygous population. But in the both the conditions, we are aiming at a single, the two aims first. One is develop the heterozygous lines, and you should attain the uniformity. This is also one of the important criteria. Uniformity. Sorry. You should attain heterozygous also, and you should attain the uniformity in the population. The two things we need to aim in the breeding for cross-pollinated crop. All the breeding methods or procedures will aim towards it. Okay. Then another nature is exploitation of heterosis is one of the main objective when you are breeding for cross-pollinated crops. And when there is a high heterosis, what are the genetic variants should be high?
Dominance
Generally,
variance.
yeah, dominance, over dominance, and epistatic variance. They are actually the major contributor of heterosis. Okay, like predominant, we will discuss. Okay, next point. Like Like normally, what is hybrids? We already discussed. They are first generation F1 crosses. between genetically unrelated parents. When you cross genetically similar parent, the extent of ectrosis will be very, very less. Okay, that's why normally we tend to cross the genetically unrelated parents and we produce an F1, then that will be released as a variety, hybrid variety. Okay, then there are major scientists we have discussed. They are called as hybrid corn makers. In USA, the group of scientists they are called as hybrid corn makers, like Ricci. Can you able to tell what is the contribution of Ricci?
Convergent breeding, something like that.
Exactly, convergent breeding. Okay? Convergent breeding. We will discuss in detail. We have discussed and we discussed how to develop inbred by using convergent method of breeding. Then Jenkins. What is the contribution of Jenkins?
Prediction of double cross performance.
Yeah, that is the one. Another one. There are many contributions. Prediction of double cross performance. Today we are going to discuss early testing. Okay, today that we will also discuss what is early testing, what is the uses of early testing. Then Hayes we discussed. He also majorly contributed. Which book? The book written by Hayes?
A professor's story of hybrid corn.
Yeah, Professor Story of Hybrid Coins. Jones, contribution of Jones.
Double cross.
Yeah, double cross hybrids. And the contribution of East and Shell. Forgotten Shell only. Okay, East and Shell.
Heterosis explains.
Yeah, heterosis concept and inbreeding depression concepts. Because of all these are actually the convergent method of breeding. It is actually aimed to develop the superior inbred lines from the already existed population. And Jenkin has given the many methods like early testing and prediction of double cross hybrids. It also helps in the hybrid breeding. And Hayes actually he also developed so many hybrid varieties in maize, and he actually wrote the book called Professor Story of Hybrid Corn. And the Jones actually identified and discovered double cross hybrid, and it's most popularized in US at some point of time. And East and Shell has contributed for the concept of heterosis and genetic basis of heterosis and inbreeding depression concepts. They're all actually leads to development of hybrid industry in the USA. That's why they're called as hybrid corn makers. Then the successful hybrid production, actually the successful hybrid breeding approach must fulfill the conditions. Which are the conditions? Can I able to tell? Don't read the slide. We discussed.
Predominance of dominance and
Yeah,
genetic variance.
exactly. Means there will be a substantial non-additive component of gene effects. The non-additive components are dominance, over dominance, epistatic. Okay, substantial non-additive component of gene effects, and also the availability of genetic diversity means Y should be high. Maximum one we will achieve. Then, there should be a availability of several genetic emasculation methods. Okay, like cytoplasmic male sterility, CGMS systems, PGMS system, TGMS systems, so that the seed production will be economically favorable and which will maximum commercial success will be possible if there is an availability of genetic emasculation mechanisms. If they are following a conventional method of emasculation method, is very very laborious and it will take more labors and more money and more time. The economic point is very very like economically it's not that much favorable. That's why there will be existence of substantial non-additive component of gene effects and availability of genetic diversity and availability of the genetic emasculation systems are three conditions three conditions which majorly used uh, important for the hybrid seed production and the commercial success of hybrids. Then what are the different phases of hybrid breeding? We already discussed and we'll summarize. Okay, first we need to develop an inbred lines. Okay. Then we had to test the inbred lines. Then we had to make the cross among these tested inbred lines. Then we had to identify which combination is very good and it is having maximum heterosis. Means combination of inbred lines then into heterotic hybrids. These are the three different phases we have to follow in hybrid breeding. We will discuss one by one. First, first basic ingredient is which source population you are using for inbred development is matters a lot actually. Either you can use open pollinated varieties, composites, synthetics, germplasms, at any heterozygous population can be used for the your inbred development. What we have to do? We have to do just selfing of an individual plants. You just take a source population, select the better one, then keep on selfing, then you develop an inbred. Okay. But what are the different procedures are available? Okay. Then what is the genetic nature of this the source population? They must be genetically broad based and also high per se performance. What it is the meaning of high per se performance?
Mean performance.
Yeah, mean performance of the particular individual, like high disease resistance, high yielding, whatever the trait may be, they should be based on your breeding objective. They should be high, showing high performance. Okay, that's why high per se performance. Then broad genetic base, they should have the more maximum variability in the source population. If you don't have the variability in the source population, then how do you select the better individual? That's why variability in the source population and the high, for example, you are taking individuals and how to evaluate, I will tell. Some individuals are very good combiners but low performing. Some are moderate combiners, high performing. Some individuals are high performing and high combiners. Okay. Then you have to choose that not only based on combining ability and also based on high performance. We will discuss later. But two main criteria you have to look, they should have the source population should have the broader genetic base and also they should the each inbred line what you are going to select, they should have high per se performance. Or anything like OPVs, composites, synthetics, germplasm can be used as a source population. You select the individual plant, you self it, and you can develop the inbreds. Then we'll see whether different methods are available for inbred derivation from the source population. One is standard method, single hill method, and homozygous diploid method. We discussed homozygous diploids, what exactly this method is. Doubled haploid methods. Okay, we are developing a double haploid, then we are using it as a inbred in the hybrid development. Then the standard method is just nothing but, for example, you have a source population. It's just like a mass selection. You select progeny select mass and combining the mass with the progeny selection. You just select some individuals, then go to progeny rows, then select within the progeny and among the progeny. But more emphasis is given within the progeny race. Then you are advancing it to the next generation. Then they select the better individual, then advance to F to F6, then you develop inbred. This is simple pure line selection. Okay. It's also called a standard method of inbred development. Then single hill method. Here what we will do, here more emphasis given between the progenies. We are comparing between the progenies and we are discarding the progenies which are not desirable. Just reject all the progenies. Select only better progeny. Then you select among them. Then here actually the sowing like per hill, we have to grow for 3 plants per hill. Then you are seeing among them. Okay, just a modification of standard method. So you will get a better visualization of the performance between the progenies among the progenies. It's just a modification of this method of sowing. Like they are comparing taking the 3 seeds per hill, then growing at a single this one, then they're comparing. It's just a modification. Then when come to homozygous diploids, They are exactly look like your double haploids. They are not look like, they are double haploids, double haploids only. What are the method we have discussed yesterday? They are the same methods which are used for development of inbred from the source population. Then other methods, these are from the original population. When you develop an inbred from the source population, but we're having already an inbreds, but due to the admixture or due to the time, there will be a contamination in that inbred that make less performing. For example, you are developing inbred for some trait, but due to time, there may be susceptible to some new diseases. There may be lower performing due to new diseases or new environmental conditions, changing climate. You want to develop a superior inbreds on the existing inbred lines. Second cycle of inbred lines. Then these methods are very much useful in developing inbreds from already existing inbred population. Means to develop a superior inbreds from the inbreds, original inbreds population. We can use a pedigree method. I think everyone know what is pedigree method. You select the better individuals, then make the cross among them, then create the transgressive, create the segregating population, select the better individual and keep on selfing and develop a pure line or a inbred which is having the complete homozygous in nature. Then the cumulative selection. Here, in a pedigree, we are going to develop a families. We are doing a selection within the family and between the families. Then we are again crossing between the families. Then we are creating again segregation. Then again you are selecting. Means you are accumulating all the desirable genes into a population, then you are choosing a selection. Then you are choosing a plant, then you are doing a selection. Means you are conducting a pedigree, then you are creating a families, then you are selecting between the families and among the families, then you are crossing between the families, again you are selecting for the superior line, then again you do for continuous selfing, then you develop an inbred. This is called as cumulative selection. Means you are cumulating all the desirable genes from into a single lines or single families from different families. This is what a cumulative selection. Then another method is called as gamete selection. Who given the gamete selection?
Adler.
Stadler. And gamete selection is nothing but, for example, you take a source of inbreds you want to develop, then you cross with the source which they may have a very good genes, genes having good lines having good genes, then you are developing an F1. You self it, then you test cross with some testers, broad based tester, test cross. Then again you cross with this one with original inbreds with the same testers which are using here also. Sorry, actually pen tab is not working. Okay. Then what you have to do? You are crossing this F1 with a test cross. Here you are getting a progenies which here you are getting progenies. If any of this progeny is outperforming this check, you regard this as a check. Outperforming this check, it hypothesizes that they are getting good gametes from this source population. Then recover those very good plants from the self seed. Then you advance into the up to F6. Develop an inbred from this. This is what the gamete selection. You are selecting based on the gamete. Understood what is gamete selection? Inbred have to cross with any good source population. Then you develop an F1. Then you cross with a tester. you cross with a tester, and along with that original inbred population again it cross with the same tester here. The progenies in the test cross is any progeny which outperforms the yield of the checks. Okay, here also check progenies are there. Means they are receiving a good gamete. These progenies, they are getting a good gamete from the source population. Here, F1 is selfed and crossed. Those plants which are showing outperforming from this check, those selfed seed can be advanced to F6 generation. This is called as gamete selection. Understood what is gamete selection?
Yes, sir.
Yeah. Okay. Then it is given by Stadler. Here you are selecting based on the gametes. Okay. Which is the gametes which are contributing more like high performance to the progenies. We are able to select them. Okay. That's why it's called as gamete selection. And it's given by Stadler. Then another method is called as convergent improvement. Convergent, as you told, convergent is improvement was given by Riche. What does it means? Actually, whilst normally inbred development has actually inbred population is inbreds are actually based on the combining ability. You have to choose for the combining ability. But if you are doing anything improving hybridization or selfing selection, it will affect the combining ability performance of the source inbred lines. For example, source inbred lines are actually previously developed based on the better combiners are there selected and they are made, they maintain their population. But if you are doing anything like pedigree selection, gamete selection, or any standard method of selection, what you are going to, you are altering the combining ability of these inbred lines. But there is a method called as convergent improvement. Without altering the combining ability of these inbred lines, we can able to develop. Like what they were doing, there, for example, in convergent development, we are selecting an inbred line. Then you are crossing an F1, like select the better inbred lines. Then you cross that inbred lines and your F1 is developed. F1 is back crossed to both the parents. For example, this is A and this is B. This F1 is back crossed to both the parents. Then what will happen? Here you select for traits for B. Here you are selecting traits for A. So that then after recovery of, then you go for continuous back crossing. Then recovery of maximum recurrent parent with some important trait. Then you go for continuous selfing. Then you release the variety. Means you are taking two inbreds and crossing, and F1 is you are getting. Then you back cross to the original inbred line, so that the nature of this inbred, like original nature of this inbred, is not altered, but you are improving for some desirable trait. It's like the same back cross. How you develop in a back cross, like final variety, it exactly like recurrent parent, but with desirable trait. Here also, the inbred, which the properties of the inbred not altered, but they are improved for some trait. How it can be done? It is done only through back crosses. Understood what is convergent improvement?
Yes, sir.
Yeah. Then another method called as recurrent selection. We have discussed in detail and different steps of recurrent, different schemes of recurrent selection for population improvement. That same can be used to develop because initially original base population will be there or inbred population. You are just accumulating a desirable positive alleles. Then you are improving that particular source population by using simple recurrent or any recurrent selection, GCA, CA, reverse recurrent, reciprocal recurrent selection. Any of the scheme you can use to develop that population as per the breeding needs. Okay. Then how to test? Okay, you have developed the inbred by any of this method, but you have to test those inbreds. How to test it? Actually, testing of and evaluation inbred is having more problematic than the test developing an inbred actually. Then normally first evaluation the first step is you have to select the inbred lines based on visual selection. Okay, based on like how they perform. Normally we take the vigor, like there when we go to the field, some the are normally the inbreds which are superior, they are having high vigor, the faster growing, the leaves are the green color. There are several traits which are by visually you can able to select which is more vigorous or which are less vigorous. That is a major. Then you are discarding some of the portion of the plants, and you are concentrating only some portion of the plants based on the vigor. But only visual selection will not able to, based on only visuals, we can't able to select the good combiners. Then you have to do top cross. What is top cross?
Open pollinated variety into inbred line cross.
Inbred into OPV. Okay, you cross your inbreds with the OPV, then you generate a progenies, then you select which inbreds are having a very good combining ability, they will definitely perform well. Those who are not performing well, you will reject all the inbreds. Then you can able to discard up to 50% of inbreds based on the your top cross results. Okay, then okay, your testing method also there, but when to test? Your F1 to F6 will be there. F1 to F6, your breeding procedure is there. But when to do evaluation? What time you have to do evaluation so that you are getting a maximum benefit in terms of time, in terms of labor, in terms of resource. Because handling thousands of inbred lines throughout this cycle, it's very difficult. It needs a land, and every cycle, every generation, it will multiply. Okay, then if you are having this much problem, then it has to do the some. You have to decide at what point you have to test the inbred so that you have to reduce the number of lines and you have to isolate the better inbreds. That is the one scientist has Jenkins has given the best method is called as early testing of an inbred lines. Okay, early testing of inbred lines. Actually, there are several questions. At some condition, at F6 evaluation at F6 will be better. At some condition, F2 will be better. Okay, but normally we will go for early testing. Means an early generation only we are testing for combining ability based on top cross. Then you are choosing a better inbred lines. Means selection based on top cross performance in early generations. That is called as early testing. Then you to identify the superior inbred lines from a group of size. Then what exactly top cross will do? Normally, if you are having get 100 inbreds, how many possible crosses it will going to get? How many possible crosses? 100 into 99 divided by 2. See, there are several thousands of crosses you need to evaluate.
High
Practically, it is
cross.
impossible. Yeah. If you are using a top cross, if you are using 100 inbreds, we are just seeing 100 inbreds. Means from the group of n with only n number of crosses. Means 100 individual is there, 100 OPs you are crossing. Just you don't need to cross all possible cross combination. That's why the important why the top cross is mostly used to evaluate inbred. This is the only reason. Instead of crossing n into n minus 1 by 2 possible crosses, you just evaluate only n crosses only. This is the one of the greatest advantages of your top cross method, and that you are doing in early testing. We can able to identify the inbreds with having good GCA. Normally, what top cross will do? It is identify the best general combiner. It is test for GCA itself. Means the inbred having a very good GCA, they will be selected in early generation only with only evaluating n number of crosses, n crosses, not n into n minus 1 by 2 crosses. Okay, understood this point? Why early testing and the top cross method is very important in evaluating the inbred lines? Understood?
Yes, sir.
Then the combination of inbred. Okay, you have developed inbred, then you have evaluated the inbred, then you have to make the cross combination of these inbreds and which hybrid is outperforming, having maximum heterosis. Then how to do it? If it is single cross, you develop an inbred, then you go for, then don't confuse with earlier slide. It is to develop and evaluate an inbred. You having a final set of 10 inbreds. Per take, you have identified 10 most best individual inbred lines. Then you have to cross among these 10 inbred lines. Then you have to select the better hybrids. Then what you have to do? How to select the inbred? Normally how to do? You have to go for some mating designs. Diallel mating, line into tester cross. Okay. You have to go for some mating designs. Which are the mating designs are available? Anyone guess?
Diallel, partial diallel, line into tester.
Diallel, partial diallel, test cross, line into tester. There are so many biparental designs like NCD1, NCD2,
NCD1,
NCD.
NCD2.
Okay, there are many methods for there. Based on what you are going to get. If you are doing in a mating designs, what information you are going to get?
We'll
Variance.
get
Variances you are
GCA,
going to get.
SCA,
GCA effect you are going to get. GCA and SCA variants you are going to get. Depends on
dominance,
different mating designs. Based on this, we are evaluating in which possible crosses which having maximum non-additive genetic variance, having dominance variance, and which lines having high SCA, which lines having high GCA. Based on that, we select the lines which having high SCA, which having high dominance, which having high epistasis. Like that, we are evaluating this inbred, making crosses in a particular mating design. Then you are evaluating for calculating, estimating the variances. Based on the variance effect, we're choosing the inbred length. Then that particular hybrid can be, that particular F1 is released for, recommended for release or recommended for further downstream testings. Okay, understood how to evaluate. But if it is single cross, it's okay. But if it is a double cross, how many number of cross you are going to get in double cross?
and into n
Formula
minus 1 into n minus 2 into n minus 3 by 8.
8. Okay, this is the formula. See how much number of lines you have to, for example, you have 40 inbred lines. How many number of crosses you have to make? Large number of classes. Actually, practically, it is not that much for recording. Recording started, huh?
Yes, sir, it's showing it's recording.
Yeah, okay, okay. Then, how many number of double crosses you have to evaluate? That's why evaluating all the double cross like cross combination, it is very, very impractical. That's why again Jenkins had developed some methods to evaluate, to predict the double cross performance. There are several methods he designed. Okay, there are majorly four methods, but only one method is majorly used. One is first, instead of taking all possible cross combination, you just select all possible six possible single crosses. For example, you're having A, B, C, D. A into B, A into C, A into E, A into BCD. Then again you just reverse cross and almost six possible crosses will come. Like B into A, sorry, B into C, B into D will come. There will be six possible single crosses if you are having four inbreds is possible. Just evaluate the performance of six possible crosses. Actually, it is formula will come like estimate plus A into C plus B into C plus B into D divided by 6. Just take the performance of these possible crosses, then divide it by 6. What are the value you are going to get? That is your predicted performance of your double cross if you are using these four inbreds. Understood this method?
Yes, sir.
Yeah. Then again, Another method is called as mean performance of non-parental single crosses. It is the mostly used. Means, for example, if having 4 inbreds A, B, C, what is the parental, for example, A cross B, for example, A cross B and C cross D. But this is the parental. Instead of A cross B, instead of C cross D, all possible single cross hybrid you take non-parental. Means which is the non-parental here?
A cross C, A cross D, B cross C, B cross D.
Yeah, 4. You just plus all the 4 combination. See, it's very difficult to write. That's why I'm asking you. Then you divide it by 4. What is the value you are going to get? That can be regarded as your predicted performance of your double crosser. Understood this method? Mean performance of non-parental single cross. And it is the only most accurate method. And it's easiest method. Only you have to evaluate only 4 non-parental cross combination. Then you mean the value. Then you are going to get the predicted performance of your double cross. Then another one is mean performance of set of 4 lines over a series of single crosses. Means actually formula I am not able to remember. A to B, A to C, A to D, then A to like this again. There will be set of inbreds for there. A to G, then again B into C, B into G, then plus again. Means there will be a set of 4 lines over a series of single. First you select your inbred, then you make single cross hybrid with some other individuals, then take the performance, then you sum up and you evaluate. Actually, I will share the formula. Easier. Actually, I didn't copy it. Okay. Then it is similar, but it is not only one method. Is the second method, which is mean performance from non-parental single cross, is mostly used. Yeah, I should have copied formulas also. Okay, I will set. Then mean performance of top process means that you are inbred cross with OPV plus, for example, you take 4 inbreds, inbred A and inbred B into OPV plus inbred into OPV, then inbreds D into OPV, then you divide it by 4. And what are the value you are going to get? That is your performance of your double cross. Third one, actually, I'm a bit confused. I will share that formula so that you will, by that looking at the formula, you are going to get. Okay. Only third, except third, you understood at least 3 methods of
Yes,
prediction
sir.
of double
Yes,
cross
sir.
hybrid.
Yes, sir.
Yes, sir.
Third, I will share the formula. Okay. This is the what I want to discuss. You understood now? We'll summarize.
Sir, can you
Okay.
explain once again that yearly testing, sir?
Yeah, early testing is nothing but you are doing top cross evaluation in early generation of this one. Early generation of while developing F2 to F6, there will be confusion. At what stage, if you do evaluation, you will get a better performance. But in early generation only, if you do selection, then it will be a better yield. Like the possible number of high inbred you are going to advance for the next generation, it will get less. It will going to be less. Okay, so that handling of minimal population will be easier than the handling of the maximum population. That's why we are going for early generation testing. And in early testing only we are identifying a better inbreds which having high GCA and high mean performance so that you advance them only for the next generation. For example, you have 1000 inbreds and if you are keep on selling the 1000 inbreds up to F6, it is very tedious. But if you do early generation only top cross by top cross, you identify the 100 individuals which having good GCA, handling this 100 individual up to F6 will be easy when compared to handling all 1000 individuals up to F6. Okay, that's why we are going for early generation testing.
Yes, sir.
Then we have discussed the breeding nature of genetic pollinated cross pollinated crops. We have discussed population improvement. We have discussed hybrid development. What is inbred development? Different steps and different steps of inbred development. There are steps of inbred hybrid development, genetic basis. Everything we have discussed. I think anything left out, please ask. Okay? Regarding breeding for cross pollinated crops. Synthetics, composites, and maize as a taking I have taken maize as a case study and I have explained how to do hybrid seed production step by step. What is the number of isolation, number of nicking, anther sinking intervals, and how to take the floral biology into consideration during hybrid seed production. Everything we have discussed. Okay, and I'm not going to discuss of hybrids of each and every crop, like hybrid breeding for pearl millet, breeding for sunflower, hybrid breeding for maize I have discussed, but I will not go in detail about all the hybrid breeding for mustard. Okay, you have to read. It's very, very important. How hybrid seed production will take place in each and every crop. It's very, very important. What is the male sterility systems available in each and every cross-pollinated crops and self-pollinated crops, and which is the male sterile sources available, and which is the best emasculation method available in different crop, which is the forced pollination methods which are available in different methods, and what is a planting ratios, male to female ratios in each hybrid seed production crop, and what is the border lines and how to keep, and there is another thing called as uh, as seed production point of view, like uh, uh, like isolation distance, field standards, seed standards, everything you need to be very perfect with each and every crop. Okay, there will be separate. I think in seed technology, hybrid seed production for the crops, there will be separate and angry notes is there. Please read that. For hybrid seed production for palm millet, there are set of production packages. Everything is very important in regarding hybrid seed production each and every crop. Okay, this will closes your breeding for cross pollinated crops. Tomorrow, I think I will start breeding for self-pollinated crops. Okay. What
Okay, sir.
happened? Yeah. Okay. Then anything I want to discuss? Anything you want to discuss? Please discuss. Okay. Yeah, I will discuss one syllabus also, so that what I have covered and whatever I have to cover, we will discuss. Yeah. Screen is visible.
Yes, sir.
Yeah. Actually, we have discussed about tomorrow. I think anyhow I'm covering this one. These epistrategies I have covered and these things I have covered. In nutshell, I have covered all these things. But DNA structure, genetic code, better actually now. Even I like you have to see the base, like you have to read from nested and summons. I will not going to cover these extra chromosomal inheritance. Like this, I think I have covered. DNA structure, functions, and replications. I will cover, but for more getting, you have to read books only. These are all beheading things. Nobody will help you in these matters. Okay? Because these are the things like DNA structure. Even I covered 10 points. In the books, there will be 20 points. In the exam, they will ask any other points. That's why normally it's very difficult to teach these things, basic things. DNA structure, there may be asked n number of points. It's not like that if I teach this one, that only asked in the exam, it's not like that. Anything can be asked. And read DNA structure and replication mechanisms, genetic code and enzyme relationship, transcription, translations. Okay. Mutation I will going to cover, crop domestication I will cover, and apomixis I will cover, incompatibility, all these breeding things I will cover. Then maize we have discussed, and rice I need to discuss, oil seeds I will discuss, pulse I will discuss, and we have covered genetic basis of selection, we have covered breeders equation, and role of plant breeding and history, and breeding for self monitored we have covered in nutshell, like overall basics are covered, but cheat breeding method I will cover again, mass selection and pure line, but if you need it, then only I will discuss because it's simple. Okay. Then breeding method cross pollinate. I think I have taken more time, like 3 or 4 classes to cover recurrent selection scheme, synthetics and composites, hybrid breeding. Okay. Then only this thing I had to discuss. I had discussed, but exactly how to derive this, I will discuss in biometrics. Then mutation breeding I will discuss. I think in this week only I will discuss. Idiotype and mutation breeding I combined I will take. It's a very simple topics I will cover in a single class. Then these things I will deal in detail. I have not covered chromosome structure and function. I will going to cover, and entire this portion I will cover. And we have covered all the markers. Any doubts in this one? Markers, first generation, second generation, third generation, mapping populations, Nils and BSA, util mapping, association mapping. Up to here I covered everything almost. This RNA-seq and integration of qtl mapping, this I will discuss. The genome projects we have discussed several genomics class, and then I had to discuss these things. Okay? And we have discussed reverse breeding, speed breeding, forward breeding, and regulation of transgenic in the Indian world. Please watch my YouTube videos. Okay, please watch video. I have covered in detail with nice presentation. Please watch the video. I will not going to cover in this class. Then breeding for this I will going to cover. Okay. Then maybe it will take hardly these are the things which will take nearly 30 minutes only. Like I will club them into single classes. Okay? Like PPFRA and plant protection conventions. Quarantine, quarantine need at least 2 classes. I should build only 1 class. Okay, anything apart from this you want to ask? Just tell only because I have to prepare notes, slides, everything. Okay.
Sir,
Bye. Good night.
quantitative genetics, sir.
I told now quantity genetics. This is the topics I am going to cover. Like quantitative characters, what is heritability, and some previously asked questions about heritability. We have discussed genetic advance, same thing only. Then variance components I will going to discuss, linearity tester, North Carolina designs, and correlation and regression I will going to discuss. Path analysis I will going to discuss. Then G interaction, it's very important I will going to discuss. And population genetics, I will going to discuss what is Harding equilibrium, structured and unstructured populations, and speciation. What is speciation? In part B, there are a lot of questions based on speciation. Okay, then what is inbreeding coefficient? This year there will be a problem on inbreeding coefficient and problem on population mean. And what is linkage disequilibrium? We have discussed in detail what is linkage disequilibrium, but some problems based on linkage disequilibrium I am going to discuss, which previously asked in the exam, like LD based, which role in plant being we discussed, and selection index I will going to discuss. Okay.
Yes.
Bye bye. Good night. Good night.
Good night.